Hello everyone, today I'll be talking about post-transcriptional modification. So what do you understand by post-transcriptional modification? It is a set of biological process common to the most eukaryotic cell in which an RNA primary transcript, that is immature RNA, is chemically altered. It basically occurs in the nucleus of the cell. This post-transcriptional modification known as co-transcriptional modification. It can be also defined as the process of chemical modification of a primary transcript to generate functional RNA product is called post-transcriptional modification or RNA processing. So what do you mean by coupled transcription and translation? Generally in prokaryotes, there is no nuclear membrane to separate the transcription and translation process, which allowing them to be coupled. It occurs when RNA polymerase and ribosome can bind to the same RNA transcript whose transcription has not yet concluded, thus leading to the functional transcription and translysis by forming a complex. There are three major steps involved in post-transcriptional modification. The first one is RNA capping at 5' end, that is 5' end capping, addition of a poly A tail at 3' end, that is polyadenylation. This is the second process, and the third one is splicing or removing the introns. So moving to the first process of post-transcription modification is the addition of 5' cap. So what is cap? The modified guanosine, that is 7-methyl guanosine, is added to the 5' end. Instead of 3' to 5' linkage, it forms an unusual base pair, that is 5' to 5' linkage known as cap. In this image, you can see that the process of mRNA capping. Let me explain about the process of 5' mRNA capping. The 5' end of primary mRNA transcript is modified by the removal of a phosphate from a triphosphate functional group. This capping process is catalyzed by an enzyme, that is guanyl transferase, which catalyzes reaction between the 5' end of a RNA transcript and guanine triphosphate, that is GTP molecule. This capping process involves in methylation by addition of a methyl group, that is CH3, to the base pair in the newly added nucleotide and to the 2' OH group of the sugar of one or more nucleotide at 5' end. In this picture you can see that this is the methylation process. Moving on to the next slide, the importance of 5' capping. This cap provides protection from ribonuclease degradation. It stabilizes the mRNA. The capping enhances the translation and splicing. It enhances transport from nucleus to cytoplasm. The second process of post-transcriptional modification is the addition of a poly A tail, that is polyadenylation. The addition of the polyadenylation takes place by the enzyme poly A polymerase. This is the picture which I'll be explaining the process of polyadenylation. The 3' end of primary mRNA transcript is modified by the addition of a poly A tail. It is catalyzed by poly A enzyme. The poly A tail is formed of 2 to 250 nucleotide residues. Before the addition of a poly A tail, an endonuclease removes a few nucleotides from 3' end of the transcript. The cleavage site has the tail recognition sequence, that is AAU, AA, and GU rich site. In this image, you can see that this is the cleavage recognition sequence. This cleavage site has the AAU AA recognition sequence and the GU rich site, and it is recognized by a polyadenylation complex. This complex is formed by joining of poly A and poly A binding protein, that is PABP, and the cleavage polyadenylation specificity factor, that is CPSF, and the cleavage stimulation factor CST. This complex binds to the tail recognition sequence, that is AAUAA, and GU rich site. It stimulates the process of cleavage of nucleotides and addition of a poly tail. And the importance, it has a similar importance to the 5' mRNA capping. It stabilizes the mRNA, it increases the lifespan of mRNA, it provides resistance to the ribonuclease degradation, and it helps in transportation from nucleus to the cytoplasm. Moving on to the third process of post-transcriptional modification, that is RNA splicing. Before splicing occurs, the mRNA is called as a HNRNA, that is heterogeneous nuclear RNA. The process of cutting out the introns from mRNA transcript and rejoining the exons to form functional mRNA is called RNA splice. So basically, what do you understand by intron and exon? Exon is the express sequence. It is a coding region, and it is any part of a gene which form a final mature RNA product. And the introns are intervening sequence. It is a segment of DNA that is transcribed but removed by the RNA splicing process, and it is the untranslated region. In the RNA splicing, the consent sequence at the 5' end of mRNA intron is called 5' splice site. It has the sequence 5' AG, G, U, AA, GU, 3', and the 3' end of mRNA intron called 3' splice site, and the middle of the intron adenine residues called branch site. This RNA splicing is mainly catalyzed by the spliceosome. Spliceosome or splicing apparatus. The RNA protein complex together to form a large splicing apparatus called spliceosome. This spliceosome has SN 5 number of snRNA, that is U1, U2, U5, U4 and U6. And the 41 associated protein, the 70 proteins that act as a splicing pattern and helps in binding the spliceosome to RNA substrate. This is the spliceosome complex. In this spliceosome complex has U1, that is 5' site recognition, and the U2 is the branch site recognition, U4 is the forms base pair complex and act with the UC. U6, U5 is a 3' junction binding of U4 U6 complex, and the U6 is the complex with U4 makes spliceosome transferase. This is the mechanism of splicing. Let me explain the process of splicing mechanism. The 5 snRNA present in the spliceosome and associated with the splicing that are U1, U2, U5, U4 and U6 that I have mentioned in the previous slide. So the binding of uRNA snRNP to 5' splice site of intron is the first step of splicing. U2 plus auxiliary factor, that is U2AF, binds to the region between the branch site and the 3' splice site. It has two units, the large and small subunits. This complex thus formed is called E complex. The splice factor, that is SF1 or BBB, in this image you can see that the BBB connects the U2AF to U1 and snRNP and form complex A. The binding of U5 and U4 and U6 snRNP to this complex converted to B1 complex or B1 spliceosome. The release of U1 snRNP allows U6 snRNA to interact with the 5' splice site. By this, B1 spliceosome is converted to B2 spliceosome. The dissociation of U4 from the U6 snRNP allows U6 snRNA to pair with the U2 snRNA and to form catalytic active site. The U2 and U6 interaction results in the removal of an intron segment. This brings the 3' end of one exon close to 5' end of another exon, both of which join together. Moving on to the next slide is alternative splicing. So alternative splicing is the process in which the multiple transcripts are generated from a single gene by a different combination of exons of the primary transcript. Generally, this alternative splicing takes place in the eukaryotes. The alternative splicing takes place in by two way. The first one is trans-splicing, by splicing together exons from two different primary RNA transcripts and by splicing out the entire exon. There are two types of alternative splicing. The first one is exon skipping. It is the most common method of alternative splicing in which some exons are left out of final mRNA. When translated, the shorter mRNA will create a smaller protein. The second process is intron retention. The alternative mRNA from a gene can be generated by including introns in the mRNA instead of removing them. In the significance, I would like to signify that the proteins are synthesized by ribosome, translating mRNA into polypeptide chain, which may then undergo modification to form the mature protein product. The post-transcriptional modification of protein, which are not gene template based, can regulate the protein function by causing changes in protein activity, their cellular location, and dynamic interaction with the other proteins. These are my references. Thank you.